Hi friends of the sun, welcome to a weekly reading group that happens every Monday, and you can join it yourself if you want. It's just on Zoom. Yeah, all the information for that stuff is down in the description, and there's also a link to our Slack channel where we just have discussions and so on, and you can vote for papers and yeah, stay updated about the stuff. And today we're talking about genome assembly. I think I came out of this conversation with Lovro, who presents this paper on using graph neural networks for genome assembly. Very inspired because it's sort of a problem that is, yeah, involves graphs. It has all these this traditional algorithms behind it that can be used there, and that the biology community has already tried to use for genome assembly. And I think if we want to use now graph neural networks for this, there are many interesting connections that can probably still be unearthed. And yeah, if someone is interested in using just graph neural networks for a scientific problem that is very inherently interesting, and then probably can lead also to some advancements that will be practically useful and impactful, then this is maybe one of the prime areas to look at and to have a think about more. And I hope that some other people can also follow Lovro's footsteps and look at this problem. But then I hope you enjoy learning about the problem as much as I did. I didn't know much about it before. So let's go and hear what Lovro has to say.
So thank you for inviting me. I'm Lovro, and I'll be talking about untangling genome assembly graphs with graph neural networks. And as I said, interrupt me at any point. So let's just jump right into it. Let's talk about de novo genome assembly first. So what is that? It's a reconstruction of a complete genomic sequence from a collection of short DNA fragments. And de novo here means that no reference genome is available. So how you can imagine this process, like an illustration, is that if you have multiple copies of the same book, that you, like all of them, you pass to a shredder and it shreds books to like tiny pieces of paper. And somehow, like, just comparing these pieces, you are supposed to reconstruct one copy of the original book. And just like in this process, some of these pieces would probably get damaged in one way or another. We are also limited in actual genome assembly by sequencing technologies because they don't usually provide you with the perfect sequences. These sequences always contain some sort of errors. Then there are different sequencing technologies. They differ mainly in the length of the reads they produce or the accuracy of them. And what's been like a popular technique for assembling is combining different sequencing technologies to get state-of-the-art assemblies. Why we are doing it? Well, there are two reasons. Like one is fundamental science, like assembling genomes of different species allows us to study the genetic diversity, evolution, their adaptation over time. Yeah.
Will you also talk about what exactly do we get out? Like, what are the snippets now? How long are they, and so on?
I can give a brief overview of this.
Okay.
Maybe at the time when I get to the actual reads and like a bit more about the process, I can mention briefly like different sequencing technologies and how they differ.
Cool.
Okay, if I forget, just remind me at a later point.
And I believe you that it's useful. I want to know what our genome is.
Yeah, so this is like there's been a lot of effort in the field of sequencing different organisms. Also, if just like for some fundamental research, like studying evolution, adaptation over time, also like knowing more about some extinct organisms. But also it's very useful for applications in medicine, like personalized medicine and targeted therapies, because you need to, even if you just want to do like a simpler, cheaper type of sequencing and map it onto a human reference genome, you need to have a reliable reference genome. And we do, in fact, have that already. There is a human reference genome, a paper that was published in Science in 2022, actually, sorry. The preprint was out the year before. about the first telomere-to-telomere, meaning like end-to-end human genome assembled. Now, you probably have heard about the Human Genome Project that was active in the 90s and like early 2000s, and that claimed that like they assembled the entire human genome, which was partially true. They assembled most of the interesting regions of the genome, but not all of them. Like if you account for all the heterochromatic and euchromatic regions, 5 to 10% of the genome was actually missing in the early versions, and there have been multiple improvements to it throughout the last 2 decades. But 2 years ago was the first time they actually assembled it telomere to telomere with what they claim to be like 0 bases missing. You had a question?
What did I want to ask? Yeah, right. I basically just want to ask what are telomeres.
Telomeres are the ends of the chromosomes. So each chromosome at both ends has telomeres, which are like some, let's call it like parts that are protecting the chromosome, the sequence to getting attached to something else.
And your book analogy before was also then kind of fitting because we, so to say, have 23 books. Each book contains a string, and we're assembling or trying to assemble these 23 strings. So
in a way, yes, but the book analogy, you can maybe imagine that each book has 23 chapters, and then each chapter would be 1 chromosome. But then the important thing here is also that you don't just sequence one. When you're sequencing a genome, you don't just have 1 copy. First, you replicate it multiple times, and then you sequence multiple copies of these of the genome.
Well, I just don't like this now to put it into chapters because chapters are usually ordered, and for 23 chromosomes, we don't really have an order right in there.
That's a good point. That's a good point. Yeah, I mean, you do have an assigned number for each chromosome. Like you have chromosome 1, 2, 3, up all the way up to 22, and then XY. But that's kind of arbitrary. Like they ordered them by length, and once it's all floating in the genomic in the sample, in the biological sample, and once you sequencing and try to assemble it, you are right. There is no inherent ordering.
Okay, so it's sort of we have a cell, or we take a cell of a human, and we know all the soup is in there, and then we take the DNA, we replicate it many times, and then we have like yeah, many of these books. Then we snipple it all up, and then somehow read the snippets of sequences because
Precisely.
our technology isn't good enough to read the longer sequences. And maybe we can get into a little bit like when can our snippets be longer, when are they shorter, and
Okay.
what the different types
So
of
that mainly depends on the sequencing technology that you're using. There have been, let's say, 3 major generations of sequencing technologies. First one is the Sanger sequencing that was used like in the it was developed in the 70s, if I'm not wrong, and was used all the way up to 90s and 2000s. The problem was that it was very expensive and slow. So it's not really used nowadays, as far as I know. The second generation is short reads. And short here means that reads are 100 up to maybe a few hundred bases long, and they are very accurate. The main company producing this kind of reads is called Illumina. They are based in San Diego. You can look it up. It's a pretty big like biotech company, mostly specializing in sequencing and some solutions for sequencing. And then like in the last 10 or so years, there have been 2 other companies which started producing these 3rd generation sequencers, which are Oxford Nanopore and PacBio, Pacific Biosciences. And Pacific Bioscience, like both of these produce reads that are longer, but of lower quality than Illumina, than the short reads. So for Illumina, they would say that the reads are maybe 99.99% correct. I think that's like 1 base in 10,000 or so that would be wrong. PacBio, their currently main type of data is HiFi reads, which are yes, you're just writing down notes, okay?
Yeah.
I'm just taking a look. Yeah, sure. I was talking about PacBio, yeah. So their reads around maybe PacBio HiFi around maybe 99 or so % correct. But they're a lot longer. Instead of like a few hundred bases, they can be around 15 to 20,000 bases long. And Oxford Nanopore are even less accurate, but can be even longer. Like they have a very wide length distribution. And in some instances of these sequencers, like the reads can be even up like more than 100 kilobases, or like they can be even up to 1 million bases long. So the currently very popular approach in assembly is combining PacBio HiFi and Oxford Nanopore ultra long reads, because PacBio HiFi are still like they are accurate enough, though not as accurate as Illumina, and long enough to bridge certain repetitive regions, and Oxford Nanopore are long enough to bridge majority of the regions.
What's the error frequency in Nanopore?
Nanopore, I think reported around 90 to 95% or so, but they've been improving it a lot lately. So I wouldn't be surprised if it's a lot better now.
Nice. Okay, that's cool.
Sorry.
And okay, well then, how long is one chromosome?
Lengths of the chromosomes differ. So the shortest ones are around 50 megabases, and the longest ones are around 250 megabases. Sorry.
50 to 200 megabases.
Yeah, so reads, I'll type it down. PacBio HiFi, read length is maybe 50 to 20 kilobases. Chromosome 1, human chromosome 1 is 250 megabases.
Yeah.
So like a few orders of magnitude longer, and not like there is no technology that comes close to sequencing the entire chromosome in one go.
Maybe that's also might take a long time then to do. If we
Yes, and I think there are. I think Oxford Nanopore is trying to develop this. They're claiming it. I mean, not really claiming. They're saying that like their next aim is to produce sequencers that can sequence some chromosomes telomere to telomere.
So and then the three places were Illumina, Oxford Nanopore, and the third one, PacBio HiFi.
PacBio HiFi is the type of reads HiFi, but the company is called PacBio or Pacific Biosciences.
PacBio. Okay.
Sorry, yeah.
Cool. Then yeah, how do we continue?
Sorry, can I ask a question? Sorry.
Yeah, sure.
Yeah, so because I am a little bit confused about the problem itself. So you pass, so going back to the analogy of the book, let's say you can only you can only sense or acquire data from short parts of the chromosome. That's why you shred it and then try to reconstruct it. So the
Correct.
problem is kind of a limitation on the biological side, where you
Correct.
cannot go. But if you were able to sense or to capture the data, or to, from polymer to polymer, this problem will make sense. Am I right?
Correct.
So
Correct.
typically,
But
so
I
from a very
don't
high
think
level,
the technology I don't think is like nowhere near. At least like maybe
okay.
Nanopore manages to sequence like some chromosomes, shorter ones, telomere to telomere. But for longer ones, I think you simply have some biological limitations, like how the DNA is degrading when you're trying to do this. And yeah, like the longer ones, I don't think anybody will be able to sequence
And,
anytime soon.
Yeah, just a follow-up question. So typically you would take the DNA, you
Yes.
would cut it, and then you would sense or acquire data from shorter lengths, and then you would try to find some overlap or something so that you can reconstruct the full.
Yes, and we'll get to this in like a few minutes,
So I
given
guess,
the amount
okay,
of questions.
no, yeah, but I guess so there are two questions. Sorry, okay, I can last follow-up question. So there are two
No, please
problems.
go ahead.
How do you cut the DNA, or at what lengths do you cut the DNA? Which I guess the longer the better, because you have more overlap, so it's easier to reconstruct. But it comes at a cost from a biological side, because it's more difficult to sense, let's say, a larger piece of DNA.
Yes, but cutting is not really done by us. It's not really done by you. It just depends on the sequencing technology that you are using.
Yeah, but
So
I guess you could send a CRISPR or something to cut places of the DNA. You can
Yeah, but
do that.
so I think using something like CRISPR would probably be not efficient enough or something for the sequencing stuff. And we now maybe have 100 copies or 1,000 copies. I don't know, maybe Lovro, you can say the number. And then we just have this technology that now goes ahead and randomly cuts somewhere. Like
yes,
Mhm.
yes.
random snippet, read this random snippet.
You could, I think, do what you suggested, like use CRISPR to cut at certain locations to get these sequences of a certain length. But I think that way you could only produce shorter sequences than what you are getting. Because if the sequence is too long, the sequencer could break it down into smaller pieces, regardless of what you provided with initially, right? And I'm not that familiar with CRISPR, but I think like you would need to know which regions you want to target, like where exactly you want to cut, right? Here you don't need to know anything. You just give this library that you prepared, and you have no idea what's contained inside. You just feed it to the sequencer, and it will produce it with some kind of data. What kind, like how accurate and how long, will depend on the machine. Okay, is that satisfactory? Okay, thanks. So as I said, yes, people assembled one human genome, telomere to telomere. And the question is then like, why are we still actually working on this? There's even on human genome, like is a human genome assembly even still interesting problem? And I would say that yes. Because if you want to use this in medicine for targeted therapies on individual level, and if you have just a single reference genome, the one that was reconstructed is belonging to like a Caucasian person from North America, and you would necessarily have some biases that would affect your variant discovery and overall the analysis of the genetic material, because we're all different and we all have different genomes, and these genomes can differ quite a lot. Sometimes up to 1%, if I'm not wrong, which doesn't really sound like a lot, but it can be critical if it's located in some interesting regions, if regions that are coding for proteins, regions that have some important structural variations. And that's like, you cannot use this kind of reference belonging to a white Caucasian person from North America and sequence everybody else in the world and then just map their data onto this genome. That's why the current trend in the field is pangenomics, where we are trying to get a more comprehensive view of genetic variations. And here the problem becomes more or less about granularity, just how deep we want to go, how many like human like populations we want to, or like different ethnicities we want to capture. And this has also like been worked on. There is a human pangenome paper that's out where they try to get like a more diverse group of people, but it's still not like entirely representative. And for example, I'm also working on one project here at Genome Institute of Singapore about reconstructing the Indian genome. So we are making the first Indian reference genome. But here it's also the problem of like, again, like how granular you want to get, how deeply you want to go for a specific group of people. Like I think the genome that we are sequencing belongs to an Indian person of like from South India, and then the question like, is that good enough for everybody else in India, from people from North India or so? I think the answer currently is we don't really know. We don't know how many variants there could exist and whether they could affect the genetic analysis. And as I mentioned, like, this is not the only thing. Like a lot of people are also working on sequencing different animals and plants. For example, there is a vertebrate genome project which very ambitiously wants to reconstruct genomes of all 70,000 existing vertebrate species, and they are not even close. If we are talking telomere-to-telomere assemblies, there's maybe a few. Like you could probably count them on your fingers. So let's now talk about the genome assembly process, which we have already touched on briefly. Like you start with one genome, and what's not illustrated here is that you replicate it, you multiply this genome numerous times. I think Hannes asked how many times exactly. That also kind of depends on the technology. Like if you're using PacBio HiFi, because it's very accurate, you don't need to fix a lot of errors simply through this sequencing depth, as we call it. So typically it's around 30 to 40x. You can imagine this as this genome being replicated 30 to 40 times, and then each copy being split into reads. With Nanopore, it's usually a little bit more because it contains more errors, and then like if you align all the reads and you do a majority vote, you can correct more data, more of these like single base mistakes if you have higher sequencing depth. For Nanopore, it's usually above 60x, often around 100x, meaning 100 copies of the genome. Once you do this, you chop it up randomly. Here I represented this with colors so that you can see like what's at the beginning, what's at middle, and at the other end. But in reality, you have no idea about the ordering of the reads. You don't know what comes from which region. In order to get some sense about it, you have this overlap phase. Overlap means that you take each read and you overlap with every other read in the sample. You are looking specifically for these like suffix prefix overlaps because they can implicitly somehow tell you like how the data is arranged, like what comes first and what comes after. From this, then you can build a graph. This is finally where the graphs come into play. And each node in the graph represents one read, and the directed edge represents a suffix prefix overlap between two reads. One question, do you see my pointer on the slides or not?
Yes, we see it. And
Okay, good.
how do we determine that something is an overlap or sufficient overlap for constituting an edge?
I think it depends on the individual assembler. So usually like there's a threshold in length, like if it's longer than a few hundred bases maybe of this kind of like suffix prefix overlap, then it's good enough and that can form an edge. And also like it usually has to be of sufficient quality. Quality, let's say like identity, how well these two sequences are aligned.
Okay.
I cannot give you specific numbers because there's no real like consensus in the field what should the numbers be, because it very much depends on each implementation and the type of data that you are using. And since the qualities are changing non-stop, yeah, it just depends. So there's a question in the chat. There are some repeating DNA sequences. Don't they interfere with the process? They absolutely do. That's basically the main problem here. How to resolve the repeats, and like whether we can find basically what would happen if you have repetitive regions, that you would get a tangle in the graph. And the main question here is if we can resolve this tangle by finding a path through the graph, which is what brings us here. That usually what assemblers would do, they would try to identify certain patterns in these graphs, resolve them, and if they cannot resolve them, then they just cut them into individual components, which are like these path graphs. These you can directly translate into sequences, and we call these contigs. But if you have multiple components, then your chromosomes would also get chopped up into they would not be single sequence. You would get different contigs comprising your chromosome. We are trying to avoid this by finding finding a path directly through the tangles. But yes, thank you for the remark. The repetitive sequences are the main problem here. And how this looks in reality, like this is an example of one of human chromosome 20, which was sequenced with PacBio HiFi data with around 35x coverage. And you can see that things can get pretty hairy here, especially like if you're using not just one chromosome, but the entire human genome and so on. And here in the middle, you can see, I suspect that this is a centromere. Centromere is like a central region in the chromosome, which are known to be a bit more complex than most of the other parts. And they tend to be repetitive as well. So this could represent the repetitive regions. Some of the less complex patterns here that assemblers often identify are transitive edges, which are visualized here on the right, meaning when A is connected to B, B to C, and A directed to C, then you tend to cut this A to C edge because you think it's not conveying any additional information in addition to the A to B and B to C.
Here now you just keep the tangles and so on just for the purpose of a message, having enough message passing happening.
Yes, correct. So usually this is a simplified graph. So this has already been cleaned of these artifacts here. So dead ends are another type here, which like when a few nodes just go into nothing. And bubbles when you have like two or more alternative paths connecting two nodes. This has been cleaned here, I think. But then there are some tangles which cannot be resolved with these heuristics. These are kind of like simpler patterns which are resolved in pretty much like every case. And there's a question about De Bruijn graphs. I did not mention them, but thanks for mentioning them because this is like one of two paradigms how people are usually resolving approaching de novo assembly. So what I'm doing is called overlap layout consensus because overlap is that part where you try to overlap all the reads. Layout is this part when you try to simplify the graph, and then in some cases, as the final phase, you have consensus phase where you try to polish the sequence that you get. De Bruijn graph is a very different approach, I would say. It requires highly accurate data. So this was a very popular approach when Illumina data was new and when it was used for de novo assembly. But it's becoming popular again because the HiFi reads are of sufficient quality. And I will not be talking about De Bruijn graphs in this talk, but my colleague is actually working on a very similar thing, just applied to De Bruijn graphs, not to the OLC assembly graphs. And our pilot study from several years ago was actually just trying to make sense of how graph neural networks work on these kind of assembly graphs, whether they can recognize the patterns that I mentioned in some very simple simulated scenarios. And the result was that they kind of can. we were able to identify these three types of patterns and then just see what we were able to resolve them with some sufficient quality. But it was just a pilot study, and there's no real sense in using neural networks to resolve patterns that can very easily be detected with algorithms. So we were trying actually to tackle different kind of cases where there are no known algorithms or no easy ways to disentangle the graph. These cases are very often solved on every assembler will have its own approach. For example, here this is a graph produced by Raven assembler, where you have this assembly graph which is literally plotted into 2D. We call this force-directed layout, and then these edges that you can see here in red, they are longer than your average edge in the graph. And then because of this, they are identified as they could be incorrect. They maybe should not belong to the graph, and then they are cut. So this is done simply from geometrically. It's done simply through the length of the edge. But it's a heuristic, and it often also is to fragmentation. It's not really a perfect way of doing it. So we're trying to actually improve on this. And since I talked a lot about the human genome that was already reconstructed and that's end-to-end and everything perfect, then how did they do it? Manually. It was a lot of manual labor from a large group of scientists who were very diligently inspecting these complex regions. They were combining different types of data, mainly PacBio HiFi and ultra-long nanopore, and just trying to make sense of what's actually going on in the genome. Yes, Hannes.
Can this problem be formulated as like some sort of shortest Hamiltonian path or so that, yeah?
Yes, very good. This ideally would be, in an ideal scenario, it would be formulated as finding a Hamiltonian path. So again, for the context, Hamiltonian path is a path in the graph which visits every node in the graph once. And it doesn't have to make a loop, so it's not like a traveling salesman. The problem here is that because of the errors in the reads, sometimes you will have artifacts like these dead ends that you cannot visit. And if you visit this, you will not visit anything else. Or if you visit other parts, you will not visit those. Also, there's a problem that if you have a longer read and then you have a shorter read inside it, then usually what people do is they just discard this shorter read. That brings some kind of differences in the coverage, and in how would I call it, in the degreeness of the graph. So in some cases you will not be able to visit every node in the graph. So it's kind of like a Hamiltonian path with some special conditions that some of the reads, some of the nodes need to be avoided.
But path, then it would be useful problem, right?
Sorry?
If we could solve Hamiltonian path problems, then it would be useful.
You're saying that Hamiltonian path would be useful for this?
Now, if we could solve the problem of finding the Hamiltonian path in a graph, then this would be useful for this task.
I believe so. I believe it would. But
So,
again, I think you would first need to identify which reads you want to avoid entirely, so that you are able to produce Hamiltonian graph. Because in the graphs as they currently are, I think just topologically,
you have to do some
you cannot do.
pre-processing, and maybe some parts have to be processed separately from each other, and so on. There's some complexity to it, but maybe it could, in principle, then be used. But
Yes.
then let's maybe continue first and get back to this Hamiltonian path
Sure.
point later.
And there's another question about what decides the length of the edge, percentage overlaps between nodes. So we have two edge lengths here. One is the length of the edge that I will use as an edge feature, and that is the length of the overlaps. So, for example, here, this, I don't know if I can draw, maybe I can try from here to here, right? Let's call this, I don't know, 10. Let's say that this is 10 something. This is one edge that we are considering here. Then this one here is shorter, so it can be maybe 5. And this is what I use as edge features. But in the case that I showed earlier, or here, this is different. So here you don't have the lengths of the overlaps in terms of bases that are overlapping. This is just based on like how the graph that is, you know, like an abstract structure is plotted into 2 dimensions. And with respect, there are some forces here that, how to explain it? It's called force-directed layout. Why? Because the algorithm tries to mimic the forces that kind of spread the graph in some way, so that it's not all collapsed into a tiny region, but also so that it's not too stretched out. That's why like the edges mimic the force, like attractive forces and repelling forces in certain way. Like I don't actually know the details of the algorithm. But yeah, this is the idea of this. And here the length is simply this, you know, like the actual length on your screen, you know, like the number of pixels or something. Okay. And to conclude this, I see another comment. I'll just conclude this, and there's we can have like a few more questions. Then the drawback, like if you're resolving chromosomes like manually, that's obviously not very scalable. People will not do this for every single genome, probably. Then if you're only using like algorithmic assemblers, they always rely on some heuristics with hand-picked parameters, and those are usually chosen by evaluating the assemblers on a few genomes, like a few more popular ones that are already assembled or have mostly been assembled, so they are of sufficient quality. And then you necessarily get a bias towards these to those genomes that you tested on, and you're not really sure how this will generalize. And then like if it doesn't, you have a very tedious task in front of yourself of like manually tweaking all the parameters of your heuristics. So we were thinking of like, if we can have a lot of data, if we have like an abundance of data that we can just give to the model, and the model can find some patterns from this data directly, that might make our task easier, and it might maybe generalize better to new genomes with enough training data provided. So the idea was, because of this, to try to use machine learning. And here is the new chapter about GNNs and how the whole setup of our approach is. But let me address the question first. And if anybody else has anything to ask about this part, like, please go ahead. You are using fixed cutoff for establishing edges between contigs, or do you normalize?
Yes, I mean,
can you please clarify?
if you, yes, that's if you have a pair of reads, okay, if you have one read or maybe one contig, and you have overlaps between well, all other contigs, for example, but you select, there are different ways to do selection here. You can select just top k, but evidently from the structure of the graph, you do not do that. Then you can select just by of a fixed cutoff that depends only on the fraction of matching reads. so sequence similarity, or you can normalize because different reads can be more or less frequent in the genome.
True, very good point. So I think there's a few things that's happening here. If we are focusing on the overlap between the reads, like how long it has to be in order for it to exist, I think that's a fixed cutoff. You know, like 500 bases. If the overlap is lower than 500 bases, then just neglect it. But there's another aspect here. Like you said, that there could be some reads that are more frequent in the read, and that's very true. So these overlap algorithms they are not usually done directly on like sequence to sequence alignment, because that would take too long. You know, like if you would do all this is approximative, all this is like heuristics. If you would do exact alignment, it would just take too long. You have millions of reads in your data set, and if reads are 20,000 bases longer, so it would take forever. So what people usually do is they compare k-mers. K-mer is or a minimizer, which are n-gram. So like it's k consecutive letters found in the genome. And there's this thing called k-mer frequency, which directly correlates with like how many, like if you have a repetitive region that's repeated several times throughout your genome, k-mers from these regions will be overrepresented compared to the k-mers found in the non-repetitive regions, right? And that's how you decide, like, from this k-mer frequency, like some of you could discard some of the k-mers, and then you don't use them in the overlap, and then you might get worse overlaps in some regions of the genome. And I think usually, like, if the k-mers are overrepresented, if their frequency is too high, they are I think they are neglected.
I see. Okay, my question was basically about if the graph neural network will have some initialization by the weights on the nodes that correspond to sequences or not. And if there is some kind of loop back on the sequences. But I understand that that's not the case, that you iterate only on the graph, and the graph is like it's given to you and you manipulate it as nodes, but you do not consider sequences themselves.
I do not consider sequences, but I do consider overlap lengths as edge features, and these are normalized, because otherwise my features could range from like 1,000 bases to 20,000 or so bases, and I was told it's not very healthy to provide your neural networks with this kind of data.
But this is a scalar parameter on the edge.
Correct. It's a scalar parameter. Yes.
Thank you.
We did have an idea of using sequences as features, like maybe embed them somehow, like through convolutions. Like you could maybe have a one-dimensional convolution that would embed this sequence into some kind of tensor feature. But I did some tests on it. It was very slow, and it didn't really seem to produce good results. So I discarded this relatively quickly, but it might be something worth revisiting at some point.
I see. It's just because this feature is obviously an interesting. Okay. Sorry. I will.
Yeah.
Yes, Hannes?
Let's continue.
Okay.
Very
Yeah, we
important.
don't have that much time left, right? Okay. Oof. 10 minutes. All right, we can do it. I won't talk about the pipeline because I'll just present everything here. The first thing we want to do is just like clarify what the objective is, right? And the objective is to get unfragmented assembly. If we want to get this, we need to know which edges we want to traverse and which we have to avoid. And for this, the network should provide us with scores that guide the path finding algorithm that we will later use. So if we just need to get information like which edges are good and which are not, then this is binary classification of edges. And this is exactly what we do. So the task is actually, the task for the neural network is very simple. And for this, we just need to have a lot of labeled training data. Since even now, but like especially at the start of this project, we didn't have a lot of reliable real training data. We used simulators. We would simulate the data to resemble the real thing as closely as possible. That way we can simulate the large amount of it, but we can also know for each read exactly where it's coming from. We remember the positions, we remember the strengths, and this then allows us that once we build the graph using like overlap phase of some other assembler, using those remembered positions, we can construct labels for the edges. Then you can imagine as if each node has start and end positions, and then we check whether edge between two nodes, whether it forms a valid overlap between two reads, whether these lead to the optimal reconstructions. If both are yes, then this is a positive example. Otherwise, it's a negative, and we are trying to avoid it. And the model finally is actually also like very simple. So like at the input, we have a graph.
That part we can ignore. But
we can ignore this.
What are the features, and that's it.
Yes. Okay. So the node features are just in degree and out degree. It's again very simple. It kind of implicitly comes just with the topology of the graph, but it was shown to increase the performance when we add them explicitly as well as node features. And like we were actually like looking for like some more informative, some more like smarter features, node features, and it's kind of difficult to find them. Because like there was no, apart from like if you want to embed the whole sequences into nodes, then there's nothing really to use.
So what happens if you embed the whole sequences into the node?
I said like I tried to use 1D convolutional networks for this, and it didn't really produce good results. But it is something that I would like to revisit at some point. I think with maybe with a little bit more playing with some tweaking, it could work better. And edge features here were like the length of the overlap and the similarity. Similarity is the identity score, just like how many bases in the overlapping part are matching. Yes?
I assume that using like a sequence embedding for every node should only work if we're training with like on the order of like 20,000 genomes, and not how many genomes do you have now?
Not enough. I trained on just one genome, actually. I would take multiple chromosomes of one genome and train on those.
Yeah.
But I don't have enough genomes to like accurate to assemble genomes to use them in the training data, because the assumption for this, like the necessary requirement to use them as a training data, is that they are assembled accurately, telomere to telomere, if possible. Okay. And the backbone of the model is GNN. So for this, we used gated GCN because Xavier was one of the main contributors on the paper, and he was working a lot with benchmarking neural networks. And this gated GCN performed like pretty well across like many different tasks. But also because it had they used it previously on a traveling salesman problem, which is something that Hannes touched on. So using gated GCN, they achieved like admirable results in solving the traveling salesman problem. And here, as I said, this is very similar problem to ours, because they also just they did binary edge classification, trying to predict which edges lead to the optimal reconstruction. And after they would get predictions for edges, they use the search algorithm, like beam search, to decode this and produce a path. So yes, this was similar to us, with one notable exception, is that in trailing salesman problem, the graphs are undirected. our side directed. So we wanted to account for this, and then we basically just like duplicated the amount of computation here. And instead of producing instead of just passing messages in one direction, in following the flow of the edges, we also would like flip the edges and do message passing in the reverse order, reverse direction. And then we would just aggregate all of this together. So this seemed like an intuitive way to do, but then it was also shown by Manuel Rossi that this is in general a good idea when you're working with directed graphs. And in their paper, they said that their GNN can be used to extend any message passing neural network to account for edge directionality information by performing separate aggregations of the incoming and outgoing edges, which is precisely what we have done, actually. But I think there were other groups, like other people who were working on directed graphs, who did the same thing, and it was kind of a reasonable thing to do, a sort of common knowledge. But this was the first paper that formally showed everything. And in training was very simple, but we noticed one technical difficulty here, that the graphs are usually too big to fit the whole thing onto GPU. And what we did was we partitioned them with Metis into individual partitions, like subcomponents, and then we would train on those. Like each of these would be one training example. What's not mentioned here, but would also help, is that instead of just like cutting edges to get the partitions, we would extend them for one hop neighborhood so that we don't actually lose any edges in this process. And in inference, we did not do this, so that we would not lose the edges that we might potentially have to traverse. Or that if we would also cut and include one hop neighborhood, then we would have we could have a conflict of, like, if you compute the same edge in two partitions, the probability for it, which one are you gonna use? Are you gonna use like mean or something? So we just do inference on a CPU. Seems prohibitive, but the model is relatively small, like a few 100,000 parameters, nothing crazy. So it's actually fairly fast, even on a CPU. And decoding, once you have the probabilities, you want to somehow get the walks out of it, right? You want to get the sequences, and to get the sequences, you need to get the walks. So here we just implemented greedy. Greedy search, it always takes the highest probability of the neighbors. We do it in both directions, like forwards and backwards. And we start by sampling the high probability edges from the graph. We don't sample just one, we sample like 100 of them for some for a bit more robustness, then take the longest walk that we find. And what we end up with is a collection of walks. These you can, if you just align them one node after another, you can translate each node into a sequence. You have the overlapping positions of them, and you can just translate walks into contigs. And that's your assembly genome. And then you can evaluate it against others with some common metrics, like number of contigs, the length of the longest contigs, the amount of genome that you have covered, NG50, NGA50, which are specific measures that I can explain in more detail. But I want to ask first if there are any questions about the previous part. Yes, Hannes.
I wanted to, can you maybe just skip through the next few slides, and I can prioritize a little bit what I would like to hear about.
Sure. So these are these metrics, and then it's just a few things about the results. How we first performed results on the simulated data, did some sanity checks, then like experiments on the real data. That was like the previous paper, and there were a few open questions after this. These questions were like, can the method generalize to whole genomes? Since first we focused on human genome, can it generalize to other species? And can it be used with other assemblers? Like if you get the graph produced by some other assembler and not the one that we trained on, can it be used in this case as well? So this was the focus of the latest paper, where we show that like even like on against the state-of-the-art assembler, we compare, we get similar or higher contiguity than this state-of-the-art on PacBio HiFi data. We identified some regions in the genome which assemblers do not manage to untangle. For example, here they cut it into 5 different contigs, 2 on the sides and then 3 in the middle here. Whereas we managed to identify a contiguous path through it that is mapped with fairly good quality to the reference. And just some results about the metrics. The paper is on bioRxiv.
Yeah.
And the limitations and conclusion.
Then let's just discuss from here on. You maybe explain NG50 to us.
Sure. It's easiest to do it with the visualization. So NG50, you can consider it as a median of length of some sort. If you have a reference that's 500 long, and you have assembly consisting of these contigs here, you order them by length, like in this descending order. And let's say that your contigs are long, like the longest one is 120, then 80, 70, and so on. You are interested in the length of the contig that covers, together with the longer ones, 50% of your reference. So not the assembly, but the reference. And
Okay.
Here you have 120 plus 80 plus 70, that's more than 250. So this would be NG50.
Then if I had an assembler that just randomly puts everything into a contig, then its NG50 would be 500.
Not necessarily, because it can randomly end up in a dead end or in already visited region if you have a tangle due to repetitive regions, and you could get lower NG50. And to account for this, like you have this NGA50 where the assembly is first mapped onto the reference.
Okay, wait
so
a
that
second.
if
So this metric only makes sense if every method has the same graph as input.
I wouldn't say so,
otherwise,
because
if
I guess
I
like the
have
graph
a-
construction is also something that you are evaluating.
Okay, but say I now fully connect my graph, and then I
Okay.
can build a single contig instead of having many contigs. And as the single contig will also be 500 base pair.
Okay, fair. In that case, yes. You would have problem with some other metrics, like the duplication rate. That's something that we used in the like when we're comparing against
Okay.
like real other genomes.
Good. So
We
this is
compare the
just
duplications and all this other stuff.
But it's sort of the main metric, like how we check the entropy is right, sort of in language models. You're getting the entropy right when modeling language. And if these base things are satisfied, then we look at the NGA50, which is sort of the quality.
Yeah, we don't have, so I guess one thing that could be considered here like this is the length of your assembly.
Okay,
If
good.
the length is too high, if it's too long, then like, yes, your NG50 could be very good, but you have assembled too much.
Yeah.
And that's a clear indication that something is wrong.
So
that can happen if you like go through repetitive regions too many times, maybe. That's like if you have an accurate reference genome that you can evaluate yourself against, people prefer to see these NGA50 instead.
Yeah. So you have these very nice results now. So are people now actually using this instead of other assemblers, or
I did get some feedback that they tried using it like to assemble some plant genomes, and the results were very good.
Can you go to the limitation slide?
Yes. Here. So I think some people tried it out, but like it's still in the it's still under the review. It didn't gain that much traction, I would say that like it became widely popular. And we're still making some improvements to it. So like, well, let's see after it's published. As for the limitations, yes, I would mention like three main things here is that if we are only focusing on layout, then we have to start with graphs produced by other assemblers, so like with other people's overlap phase, which means that like if these graphs contain certain errors, we cannot fix them. We cannot add new edges to the graph if they don't already exist. We can only choose which edges we will not traverse, but we cannot introduce new ones. The second problem is that we are currently unable to do phased assembly, and we cannot integrate multiple sequencing technologies into the pipeline so easily. Phased assembly means here that each of us has two copies of genome, one from mother, one from father. And like what people nowadays are aiming for is that you combine multiple sequencing technologies to get both of these copies, maternal and paternal. We currently just get one copy, or like you get two copies, but kind of fused together. And in the like when we evaluated on the data that we evaluated, like it's all haploid genomes, meaning they only contain one copy. So it's some kind of special cells or inbred organisms that have two copies, which are largely the same. But yeah, we cannot do diploid assembly. Yes.
So your third point here is that we can't really generalize to other genomes because we don't have other genome training data. So then can we fix that by instead only training on synthetic data and only training on okay, let's only train on synthetic data, and then maybe let's just try to formulate this somehow as a Hamiltonian path finding problem. And I think for Hamiltonian path problems, we can also obtain synthetic data, right? And then we could, I don't know, maybe string together a few Hamiltonian paths to come up with the final optimal path. What do you think of a direction like this? And then this would be much more widely applicable.
It might work. I'm not sure if it would be prohibitive in terms of computation. Because for the graph that I'm dealing with, for example, the CHM13 one, like it was, I think, like 2.5 million nodes and 18 to 20 million edges. So like we always tried to keep things very simple so that the compute power wouldn't explode on these kind of graphs. Yeah. Like we used greedy search and not beam search. Like I tried to do it with beam search, but it would the compute took forever. The amount of time that I would need to decode with beam search.
Yeah.
So okay. I think that would be my main concern, even though theoretically it sounds sound. It sounds good.
Yeah, but then there are maybe tricks that one can do of splitting the thing into subgraphs or something like that.
Yeah, something like that might work.
Like if someone is interested in machine learning or in graph neural networks and wants to work on a like scientific application, then this sounds to me like an extremely interesting direction. And like there must be so much you can do here and so many complexities that you can still resolve. And it sounds like it might also be promising. Yeah.
I agree with you. And like actually like the last point in this presentation is that there are still many problems left to solve. Like I think this is not even scratching the surface. So yes, there is a lot of work here that could be done. So if anybody is interested in this, like feel free to reach out.
And right, there are so many connections to existing work in algorithms or in graph algorithms, and there's also already all the connections that the assembly people have unearthed when trying to solve this problem. Yeah, then
correct.
Combining this with machine learning, I think you're really just scratching the surface here with this
correct
approach. yeah, I'm sure there's certainly something interesting can be done. And it seems to me very likely that something can be done that is also very useful and more generally applicable than this, where we need to still train on some human genome data.
Yes, I agree. I think there's like room for improvement on this front as well, on basically every front. But yeah, we always say like it's, you know, like for some future work, because like we're also on one side we're limited with data as well. So the more other genomes are assembled, the more data we'll have to train, and the more data we'll have to evaluate on of like how generalizable this thing actually is.
Yeah, I'm hoping that we can get away with only training on synthetic data and can use all the data for evaluation.
Yeah, that would be great. And I was trying to, I mean, I did some tests of including some like small amount of human data that we had for like fine tuning or so, but the results were like, pretty similar to what we're getting just with like the large amount of synthetic. So I think it's still like we still have to see like how exactly approach this. But the training on synthetic data, like this approach is very clean because you have the positions for each reads, and you know that you are not mislabeling something. There's also the whole thing that we touched on, like the De Bruijn graph approach. So like there are several like very good assemblers that produce very good like assemblies that work in a completely different paradigm with De Bruijn graphs. So that's also like an interesting direction.
Okay.
Dennis, you have another question?
Yes. My question is about data synthesis. How close it to the actual genome in terms of the latent factors that you actually consider? Maybe you can. So my question, I guess, is that perhaps to get better results, you can do adaptive usage of sequence embeddings for the nodes that are challenging. Essentially starting with the setup that you have, with nodes initialized only by degree of in the graph, but then in challenging situations, maybe considering sequence themselves. Because I believe that, yeah, there is an additional complexity that you have multiple chromosomes, and perhaps between chromosomes you can have connections of duplicated genes, and then if you want to do phase assembly, it's an additional headache. So maybe knowing some implicit understanding about distribution of the DNA sequence can help. Yeah, and hence my question: how faithful is the synthetic data?
So that's a very good. Like all the suggestions are very good. It's something that I haven't really tried to do, but yeah, like it sounds, it could be promising. How faithful is synthetic data? I would say fairly, because so like we took the exact distributions that we took the exact length distribution produced by PacBio HiFi reads and simulated and used this distribution for our simulated data. We also took the quality scores of PacBio HiFi reads to use in our synthetic data. The problem here is that there could be a discrepancy between the quality scores that are reported for the bases and the actual quality. These quality scores, they're produced by assemblers or base callers who translate the signal into the reads. And they are somewhat like some kind of estimations of how good a base is.
I see. So yeah, I just thought that you actually started with the DNA sequence, but you started with reads and not actually even reads, but kind of like abstracted reads, right?
We do start with the DNA sequence, but using these, using the length and the quality distribution, we sample the reads from the DNA sequence.
Okay. Sorry, Van, for
So yeah,
misunderstanding.
no problem. So these distributions simply dictate how we will sample the reference, but we still start from the reference, the DNA sequence.
Yeah, I was thinking more about the reference. Then maybe last question: when people assembled this human TDT and telomere-to-telomere genome, and you said that it was done, quote, manually. Did they also
Yes.
rely on actually the sequence itself, not just on the distribution of like overlap scores? Did they also consider, I don't know, like maybe some known clinically significant repeated regions, or maybe something about known repeats of a human genome that go from like retrotransposons and so on?
Yeah,
Or
I'm
was
pretty
it
sure
not
they
taken?
did. I'm pretty sure that like when they encountered a problem of repetitive reads in a certain region, they knew exactly which region that was, and that they used some, like biological insight into what should actually be there, like how many repeat units should maybe be located there. For example, one notorious region, or like notorious regions, are rDNA arrays. rDNA arrays it's a tandem repeat where you have like some kind of pattern that's just repeated one after another several times, and you don't really know how many times, especially not if you're trying to do de novo assembly. So I think how they estimated the number of these copies of these rDNA arrays is by using some kind of fluorescent tagging, that they would tag these regions and then try to like visualize them, and somehow, depending on the intensity of like how shiny these parts would be, like what's the approximate number of copies that each region has. So like they knew that they were dealing with rDNA arrays, that those were the regions not resolved, and they knew how to tackle these problems.
Yeah, maybe last idea. I'm not quite familiar with the distribution of different repeated sequences in in many genomes, but even in human genome. But maybe if they sort of have low complexity and they have a relatively limited evolutionary set of source distributions, such as viruses, like retrotransposons and so on, then maybe sort of this mapping between repeated regions, it can be both inside a repeat of a single kind, but maybe also between two different slightly altered families of repeats, if my intuition is correct. And then maybe it is interesting to abstract this away and just look at statistics of the repeats themselves and just compare different types of repeats, how they can overlap, which ones are similar, which ones are dissimilar, and maybe this can help somehow.
Yes, that's interesting. I don't think I can tell you more about it. I don't think I have this biological knowledge about the repeat regions of different organisms and how translatable they are from, let's say, like viruses to human and to other species. But it could be, it's an interesting approach. It could be something that would be, a nice thing to implement into the model if we could somehow like recognize which type of repeat we're dealing with, and if we can take the knowledge from other species to resolve this one.
Yeah, I actually meant viruses like inside the human genome with retrotransposons, those types, yeah. But if you want to do a general system for many different species, this can be complex because there are probably different patterns in different species, but I'm not that familiar. Thank you.
Yeah, no problem. Thank you for your questions.
So now I'm no longer muted. Good. Then I wouldn't have any other questions, so let's call it a day here. Thank you,
Sure.
Lovro, and thank you for what am I thanking you for? The nice paper the presenting the thing, and I think inspiring me. I find it very inspiring that problems like this exist, and yeah, hopefully some other young graph neural network aficionado will join you on this journey to join you on this journey to use GNNs for genome assembly.
Yeah, that would be great. And I thank you very much for inviting me. It was a pleasure to present here, and thank you everyone for all the questions.
Yeah. Extremely cool problem. Thanks for explaining it to us. And see you around.
No problem. Thanks for listening, and yeah, see you all.
Bye, friends of the sun.
Bye.